and get this meeting started. All right, so welcome everybody. Thank you for being here to our July monthly meeting. It's very exciting to have everybody here today. So just a brief introduction. So we do not have any field trips currently planned. Our next field trip will be in, our field trips will resume in September. So stay tuned to our Facebook page and our website to see what's coming up. Everybody's been working really hard to make some, make this next round of field trips as successful as our last. We do have some volunteer opportunities within the organization. Some of the featured positions are listed on the screen, you know, a volunteer coordinator, community outreach. But if you're interested in volunteering at all, just reach out to us at the email address on the screen. It's info@auduboneverglades.org, and we can share information with you all if you're interested in volunteering about what we have going on. You know, we're trying to make some changes and kind of liven things up. So hopefully we can get a nice volunteer base to get that going. A reminder to everyone, please renew your memberships. So it's time to renew for the June to May fiscal, or not fiscal year. My mind is at work. The June 1st to May 31st, 2023 year. So if you do need to renew still, you can go to our website, and then right there on the home page, there is a box that lets you become a member. So it should be highlighted now on the screen, a blue box. You just click on that, become a member, and you can renew your membership there, or renew or become a member if you're not a member yet. So please join us. The photography group, the 2022-23 photography group programs and activities will start in September. Make sure when you register with your Audubon membership that you join or renew your, to register to be part of the photography group as well. So all Audubon Everglades members can also be part of the photography group. For more information, you can go to the website. It's linked on our Audubon Everglades website, so you can get there from the auduboneverglades.org, or you can go to photography.auduboneverglades.org. Our speaker next month might look familiar. It's me. I'm going to be presenting on work that I do as a wildlife biologist at the South Florida Water Management District. Not going to talk too much about that because I don't like to talk about myself. And then we do have a YouTube channel, so please check it out. You can, you just go to youtube.com, you search for Audubon Everglades, and then you can subscribe to our channel. You know, when you search for Audubon Everglades, it pops up. It's the cartoon of the snail kite right there. And then the yellow arrow on the screen is showing you, you just hit that button to subscribe, and then you'll be able to see all the videos. So all of our presentations are on there. So if you ever miss one, you can go back and watch them there. I know a lot of people work very hard to make this happen. So go ahead and check it out.
Is it our YouTube?
So just a reminder during the meeting today, if you have any questions for any of our presenters, please put them in the chat box and we will go through the questions at the end of the presentations. Additionally, please make sure that you're muted during presentations. We do, the hosts and co-hosts do go through and try to mute people, but it's a big help if everybody who's not presenting or speaking is muted at all times. And with that, it comes to our lovely Bird of the Month presentation by Clive and Cece. So I'm going to go ahead and stop sharing my screen. And then Clive can share his. Or Clive, you should be able to just kind of start your presentation now.
Okay. Okay. The new share.
We saw it for a second.
Yeah. There we go.
There it is.
Yeah. All right. Well, hello, everyone. From Climate CC, we have been looking forward to presenting this particular bird of the month. I just want to take a little bit of a segue to mention that if you hear me sniffling during my talk, allergies are attacking me again. So I apologize in advance. Our bird is the great kiskadee, and it was a great pleasure for me to see this bird for the very first time several years ago on my first trip to Mexico. I was actually there looking at some of the Mayan ruins, and there was a forested or wooded area nearby, and I saw a flash of gold go by. I glanced over, and lo and behold, there was a kiskadee. Not only just in tremendous plumage, but more times than not, you hear them before you see them. They make an absolutely raucous call, and several times, several of these birds will actually join together in a chorus, declaring who they are. Their names, the name Kiskadee actually is derived from the name, from the call that the birds make. And if you guys have ever had the opportunity to listen to them, which I'm sure you can, if you have the Audubon app, you would actually be able to hear what these birds sound like. Just wanted to mention, someone has their microphone on and I can hear them. Okay. One of the amazing things about this bird is not only the vibrant colors that you saw in the initial photograph, but the top of that crown that looks like it's all black has a hidden yellow explosive plume of feathers that actually comes out. It looks like the bird's actually blowing his stack. They actually display this yellow when they're in an aggressive mode towards another set of kiskadees, like two males in an aggressive mode. But the male will also demonstrate this during courtship as part of the courtship ritual. The bright yellow breast, white throat, the black going through the eyes, those warm brown colors on the wing are just all work together to make this bird spectacular in its image. And you'll notice there in the photograph on the right just how absolutely gorgeous the bird is. In flight, you get a chance to see a lot of those hidden colors in the underside of the wing and tail that are usually covered up when the bird is standing on the perch, but just gorgeous bird. The range of the bird is pretty interesting. My research showed that they're in eastern Texas, southeastern Texas, but they do occasionally get up into New Mexico, Arizona. One was even in Louisiana at one point. And as you can see, there's a vagrant over there in Florida to the right side of Lake Okeechobee. However, the bulk of the population exists from central Mexico, central America, all the way down through South America to Argentina. So a pretty extensive range for this bird. It's quite conspicuous in the few locations we have the bird seen here in North America. However, there are so many other flycatchers, of which the bird is a member of the family, the tyrant flycatchers, and so many other flycatchers throughout Central and South America are so close in field ID marks that it makes it challenging throughout those areas to distinguish the great Kiskadee from several of those other flycatchers. Here's an example of just some of the habitat preferences that the Kiskadee has. We have the tropical deciduous forest. Throughout Texas and Arizona, places you would find them in thorn scrub habitat. They also like streamside thickets, coffee plantations, banana plantations, citrus plantations. They do tend to gravitate to areas that are open woodland areas, and in particular, those locations that are close to water. And I mentioned that because once we get to their feeding and their feeding habits, it'll sort of highlight just how special this bird is among its other relatives as a flycatcher. Speaking of which, their diet is actually quite large and varied. Just like flycatchers, as you would expect, you would see them sitting on the tops of trees or shrubs or the outer skirts of those areas, flying out and catching insects in mid-flight. However, they are quite prone to plunging kingfisher-like underwater to get small fish, or hovering above the surface of the water to snatch fish, floating insects, tadpoles, and things of that sort off the surface of the water. Your eyes are not playing tricks on you. The lower left-hand photograph is a Kiskadee holding a bat. Again, it's amazing the varied diet of these birds. They will consume, in addition to some of the critters that I mentioned before, they'll take small mice, frogs, tadpoles, lizards, snakes. They'll even take baby birds of other species from their nest as well. In addition to aquatic critters, they'll take aquatic snails as well as tree snails. And as the lower right-hand photograph indicates, they do feed quite a bit on berries and other types of fruits from vines and shrubs and things of that sort. The nesting information on the bird is sort of sparse. Once a pair is established, they are monogamous for the rest of their lives. And so the mated pair will actually stay together in nesting and non-nesting seasons. They do participate in making a very bulky nest, usually about 10 to 20 feet above ground, usually in the crook of a tree or in a lone tree along near the edge of the forest. The nesting material that's used are grasses, twigs, Spanish moss, weeds, and other plant fibers. The size of the nest is pretty impressive. Weaving all that material together, the nest actually, the finished nest is about 13 to 18 inches tall, 10 inches wide, with the entrance to the nest on the side, as is indicated in the lower right-hand photograph. 2 to 5 eggs are laid. The eggs are generally creamy white with dark brown spots or lavender spots mixed in. Both male and female incubate the eggs for about 15 to 22 days. And the chicks that, once they're hatched, are quite helpless, very little down on them, blind initially. But the nesting stage of the babies only lasts for about 10 to 12 days, and then they're out of the nest, clambering around on nearby branches. And so that's just a quick and dirty shot of the great Kiskadee. Fantastic bird. We know for a lot of us that take advantage of the summer months for traveling, if you guys are in the Texas region, definitely look for these birds. They are spectacular and well worth even going out of your way a little bit to get to see them. So by all means do so. All right, hope you guys enjoyed the presentation, and we'll field any questions anyone has.
Thank you, Clive. That was great. That was really interesting. So anybody has questions, go ahead and put them in the chat. So Paula says that she saw these birds in Mexico and they have a really great call.
Yes, they do. It's quite loud, and they do draw a lot of attention to themselves. It's interesting, as I did some research on them, especially in regards to their diet. They, for as aggressive as they are with others of their species from time to time, they actually show very little fear of humans, and quite often they will gravitate to where human activity is to hang out, to get scraps of food, handouts from, and they'll also take food from bird feeders that are set out to attract many different species of birds throughout those areas. And they're not, it's not out of the realm of them stealing cat and dog food from the bowls of these animals that are left out. So
That's cool.
yeah.
If you want to stop sharing your screen, Clive, then we can see your face while you're answering questions. We do have a question from, oh, they all went away, hold on. From Janice. She would like to know, so I guess she's asking what species or who would bother their nest?
There are mammals, larger animals that will try to take advantage of them. There are snakes that will try to get them, other types of raptors that will try to get them as well. So they are pretty aggressive. Both a male and female will gang up, if you will, and attack any of the potential predators of the nest, but they will also from time to time call in for recruits to come and assist them as well.
That's interesting. So by recruits, do they have like family groups? So is it like the young of last year that help?
That's exactly right. Yeah, you got it. Yeah.
That's really, I love those kind of dynamics in birds. It's
Yeah.
really interesting. Jeanette says that she saw some last year and when she stayed at a resort in Cancun. Marie saw some in Costa Rica. So Gwen has a very good question here. She says, how large is the great Kiskadee?
They're actually 9 3/4 inches. So they're about a little bit larger, I think, than our mockingbirds. But what makes them stand out is the bulky stature of the birds. Their heads, their necks, the impressive colors, all of that works together to make them look so much bigger than a mockingbird, as I said, in comparison. So yeah, they're pretty impressive. And of course, that attitude of theirs, yeah, they back it up pretty well.
Very nice. That's interesting. So if anybody else has any questions, go ahead and put them in the chat. Oh, Kevin would like to know, he says, I assumed the bird behind me was a social fly catcher. Is it? I assume the bird behind me. Is there a bird behind you, Kevin?
Yeah, I do see it. It could be a Kiskadee, depending on where he was. Kind of straining to see the photo, but just
Belize. It was in Belize.
it was in Belize. It is quite possible that it was a Kiskadee. They're also very common in Bermuda as well, and they were introduced to Bermuda in the 1950s, and the population has grown tremendously. Yeah, I'd say that that could be a Kiskadee. Kevin, about how large was that bird? Just approximate.
Pretty small. I mean, so like
Nothing like 9 inches, as I mentioned, for the Kiskadee.
maybe about 7.
Okay. It could very well be one of the other flycatchers, because as I mentioned before, they stand out very easily here in the United States because of the limited range that they're in and the vibrant colors. However, throughout Central and South America, where the population is huge, and there are so many other flycatchers that closely resemble them, it could make it pretty difficult and very challenging to ID them. So color alone is tough to go by. Size is gonna play a bit in helping to identify. And of course, the calls and behavior of these birds. If you see a bird like that diving, pretending to be a kingfisher, then you've got a kiskadee on your hand.
Yeah, Frank says that the small bill size kind of points to it being a social flycatcher.
Right, yeah. Yeah, Kevin, once you brought the photograph up, yeah, I was able to see that a little better.
All right, Clive. Well, I think that's all the questions for right now. If any pop up in the chat after we're done right now, maybe you can just answer them in the chat for folks.
Sure. It was our pleasure. Thank you guys so
Yeah,
much.
thank you so much.
You're welcome.
All right, so with that, I'm going to try this whole screen sharing thing again. And I'll go ahead and hand it over to Autumn, who's going to be introducing our speaker this month. And then we'll hand it off to Dr. Dorn.
Hi, everybody. I am here despite the COVID. And I am really excited about Dr. Dorn today. So let me tell you a little bit about him before we talk to him. He's an associate professor in the Department of Biological Sciences at Florida International University. And he's also part of the FIU Institute of the Environment, where he leads a large lab of students, postdocs, and technicians studying wetland ecology. He received his PhD from Michigan State in 2003, and he's been studying the ecology of the Florida Everglades and the adjacent wetlands for more than 19 years. And he's best known for his work studying the ecology of freshwater crayfish and food webs, but his work is diverse and includes examinations of diets of nesting and wading birds. And he's also been a contributor to the annual South Florida Wading Bird Report. He's a freshwater community ecologist, and his particular interest, I think, is what he's going to talk to us about today, which is what effects wetland water levels are having on the food webs and the nesting conditions of our wetland birds, our wading birds and everything. So I'm super excited to hear about this. So with that, I will say, Dr. Nate Dorn, take it away, sir.
Thank you, Autumn. Let me pull everything up. Hopefully this is okay. Autumn, I'm going to assume you can see that screen.
We can see it. Yep.
Yep, we can see it.
Okay.
We got it. Perfect.
Great. Well, I really appreciate this invitation to give a talk here today, and it's really impressive how many participants you're getting. I can only imagine how fun some of these meetings probably were before COVID, and I assume that they will be even more fun when you guys are meeting more regularly and at your events. So. I'll just say it was very interesting to hear about the kiskadee. I did not know that, and I admit that I'm not the birder that anyone is here, but it's very interesting that they dive like that. So thank you for that educational point. Today, as Autumn says, I'm a freshwater ecologist, and I want to talk to you about sort of a book report of work that I've done in the Everglades, and as she said, I've been here working on some of these questions since about 2003. I think some of the ideas kind of go back about that far. And I will start, though, by admitting that I study mostly fish and crayfish and food webs and predator-prey interactions, and I'm not a real avian ecologist. So however, there's a number of people I just want to get out of the way right now and say thank you to, because they've sort of brought me along to understand avian ecology better. Mark Cook, who I think is frequent to this group and may even be on the call today. Dale Golick, a longtime colleague at FAU, who He's now moved on to Texas A&M. So one of the premier wading bird ecologists in the USA. Some of his students, Garth Herring, and then some of my own students and other colleagues who I've worked with who are far better birders than I am and better avian ecologists as well. So thank you to all of them. With that said, I'll get into this talk just a little bit here. So as an ecologist, I'm really interested in this translation from environmental variation to what an ecosystem does, or what sort of community it produces. And that arrow right there, I think, is kind of what we all do naturally. And as birders, and I know that you do it too, because you do this sort of translation before you go out in the system to try and understand what I might see. And so there's natural models that we work with in our head about how we think the world works. For the quantitative ecologists, we have to try and put numbers on some of these things and then work through a series of problems to translate what environmental variation means for ecosystem functions. So in a wetland, like the Everglades, and this is a picture of Everglades National Park from the air, and obviously tree islands, sloughs, the deepest water you can see there, and then sawgrass habitats as well, where the emergent vegetation. These kind of systems, a dominant feature of their environmental variation is whether or not the site dries and exactly how deep the water gets. So we're going to call that hydropattern or hydrological variation. And so how this very large system creates a lot of food for a lot of seasonally nesting wading birds is the sort of an overarching question. And we're going to call that the ecosystem function that I'm primarily interested in. So moving from the general operational mode for the ecologist, from environmental variation to ecosystem function, if we were to make it specific for the Everglades, we would say wading bird breeding activity might be a function. We want to try and understand what causes it, and it's something the Everglades clearly is capable of, has been capable of, but we need to understand how to translate at least hydropatterns or hydrologic variation into wading bird breeding activity. And particularly if we want to the system or rejuvenate the birds in the system, we need to know what kinds of hydropatterns will create better conditions for wading bird breeding activity in particular times and places. Now, birds, as you probably all know, many birds, or most birds, are going to have their breeding activity somewhat or completely food limited, given the right season and the right vegetative structures. But in the Everglades, because we can manage water levels and because we've changed the water patterns so much and the size of the landscape, trying to rebuild the landscape or reconfigure or replumb South Florida for better hydropatterns for wading bird breeding activity is an important endeavor. Okay, so if we were to just, I want to talk a little bit about conceptual ideas about how this happens in a wetland for a moment. And if we were just to follow one part of this wetland and just focus in on it and think about the different parts of the hydro pattern that affect the availability of food for a foraging bird that's going to feed to its chicks. And we focus in on one little part of this marsh here in the yellow box here. This would be a slough habitat in Everglades National Park. And if we just put the hydro pattern of this location in time, what we might see is something like this. They're on here just because I know you want to see birds. Actually, that's not true. It becomes important in a moment. But what's on the bottom is what I want you to focus on for a moment with me. So the water depth we'd see moving through the months of the year with a familiar rise in water levels during the wet season when it's raining, from May to June to July to August to September, October. We hit the peak water levels in October, and then we start to fall. And my animation already jumped up here. You can see that as we approach January, February, March, the water levels in that location that we're focusing on in the Everglades begin to approach appropriate foraging depths. This time of the year is what we sort of think of conceptually as making prey available. And it also coincides with the seasonal nesting of the wading birds, as most of you probably know. Those foraging depths at that particular location need to reach somewhere around 30 centimeters water depth. And it can differ a little bit between species, but primarily that's because of leg length limitations of the birds to stand in the wetland, or bill length limitations if you're an ibis or a stork and you feel your prey. Okay, so making prey available both uncovers the animals because they were basically in water too deep to feed on back here, but also concentrates them to some degree across habitat structures from the sawgrass habitats down into the sloughs, the deeper habitats, and then into shallower and shallower pools at the very end of the season. But prior to this, there's another sequence of hydrologic conditions back here, which is where prey are produced. So we'll call these antecedent to the nesting season or coming before it. And therefore, the conditions back here, we really want to know is what are the conditions that are happening back here that are making more or less food that then gets uncovered in the blue section over here on the right-hand side of the graph. So what I'm going to do today, though I'll mention some of the other wading birds, I'm going to focus on the white ibis, which in Florida, not the most, maybe the most exciting bird, though I still like them quite a bit. It is and was and always has been, as far as humans are concerned, and as far as we know, the dominant freshwater wading bird, long-legged freshwater wading bird in the Everglades. Today, there are 50 to 70% of all the nesting wading birds in a given year, and they, as far as we can tell, always have been. Of course, they've suffered about an 80% reduction post-drainage or in the 20th century. Now, among the Everglades restoration performance measures are these three things. First, one thing that we'd like to see is an increase in white ibis nesting numbers to sort of reverse some of these losses. Second, a return of most of the white ibis nesting to the southwestern coast of Florida. And then third, though not quite as operationalized in the goals, but to support eruptive dynamics or nesting dynamics, the formation of super colonies with increased frequency, which has been only seen 1 or 2 times since the 1930s. Now, the white ibis, I'm just going to go back here for a moment, in case it's familiar, not familiar to you. Of course, it is thought to be a generalist feeder, and obviously you see it in your lawns feeding this time of year, and is fed by your neighbors probably. But it also is known to be a nomadic species, not undergoing any particular migration, but moves around the southeastern U.S. and the sort of the foci or focus in a given year of nesting activity can move over a series of years from one part of the southeastern U.S. to another. But the Everglades was historically a stronghold for this species. Okay, so taken in historical context, then in the 1930s and the early 40s, there were this is sort of when actual records began of ibis nesting in the 30s and 40s. These are total white ibis numbers in the Everglades, and these are in nests or pairs of ibis nesting. So historically, we saw particularly in the coastal parts of Everglades National Park, upwards of 100,000 nesting pairs, and these but today we see almost all the nesting happens in the central and northern Everglades, and the long-term average up through the early aughts was about 15,000 to 17,000 birds, pairs of birds. So when we talk about coastal nesting ibis, and this is a aerial of southern Florida, picking up mostly Broward, a little bit of maybe a little Broward and Dade County for the most part here. There are about 7 principal colonies where the ibis will nest along with other wading birds in mixed colonies in the southern part of the system. And there's also Perotis palm, which I did not list here. But these have been fairly regularly used for the last several decades. But only 1 or 2 nests typically pops off in a given year. Instead, what we have today, if you can see up at the top of the screen, I'm not sure if that's showing, there is the center of the nesting, the epicenter of the nesting is primarily in the central or northern Everglades, northern water conservation area 3A, or even up into Loxahatchee, which is off my screen up to the top. So there's very little nesting happening in the coast today. And so let's just think about some of the uncertainty about this performance measure for Everglades restoration then. Peter Frederick, again, a real avian ecologist at University of Florida, set out some of the uncertainty in his paper in 2009, basically about this translation. What he didn't call it that, I call it that, but two points of uncertainty were the relationship between crayfish and ibis nesting, because although there were just a few diet studies that have been done, but in other parts of the country, and particularly the Southeast, there seemed to be some relationship between crayfish production and ibis activity, but it wasn't quite clear in the Everglades, and they're known to eat fish as well. And secondly, though, the importance of flood and drought sequences on ibis nesting, and I'm going to get to that in just a moment. These were two unknowns as of about 2009. So the first question is, what prey are being fed to nestling ibis, or in other words, what kinds of prey or types of prey fuel good ibis production or good ibis nesting? And so a few of our options are shown here at the bottom. A couple small bodied fishes that are common to the marshes of the Everglades, the mosquito fish and the bluefin killifish, and then the slough crayfish on the right. There's a handful of other species that could be fed or are fed to ibis and that the Everglades produces, including freshwater shrimp, the grass shrimp, and a whole bunch of other species of fish, including some that reach slightly larger body sizes. So I'm just going to start with one little data slide on the diets of ibis from Loxahatchee National Wildlife Refuge from one study. And this actually includes the two other wading birds in the system. These are prey, biomasses of prey fed to white ibis chicks and two small species of heron chicks, the tricolored heron and the snowy egret, from the same colony in Loxahatchee. And it was a particularly good year of nesting for all three species of birds in Loxahatchee. And so we have, but notice these birds with very similar depth requirements in terms of what depths they can stand in a marsh, they're seeing the exact same wetland, and they're feeding from the exact same central place foraging here. They bring back their food to the chicks, and they're bringing back completely different prey. So the snowy egret and the tricolored heron, both sight feeders, are feeding on fish, both sort of body size classes, larger and smaller fishes. And the white ibis is feeding its chicks almost entirely crayfish, with a little bit of garbage thrown in there. It's up by Loxahatchee. They can reach some places that some urban environments as well, though it was always sort of a minority of the diet. Now, as we look across a number of colonies that we studied from 2006 to 2009, we see that crayfish, particularly the large colonies, crayfish make up 45 to 75% of the prey biomass, and in all those cases are the dominant prey item fed to white ibis chicks. And in particular, there's one species of crayfish that we're talking about here, and this is known as the slough crayfish. It's found in the central portions of the Everglades, and relatively longer hydro periods, longer inundation times is where this species is found. And so this at least gives us some information about prey that are supporting white ibis, where the ibis are currently in the central northern Everglades. So if this is the kind of event we want to see more of, then the question is, how do we get more of the food and then to enter into the gut of this animal, which can be then fed to the chicks. All right. Now, this led to the second point of uncertainty and a hypothesis by the same Dr. Peter Frederick and John Ogden, who wrote in 2001 in Wetlands. They looked back at the historical record of white ibis nesting, including all those years in the 30s and the 40s, and they noted something. They said, look, this observational pattern, really important for ecology to have observational patterns with some data behind them. And they said, look, these really big wading bird nesting event years seem to be associated with severe drought. Now, it wasn't always very closely measured because we didn't have the water monitoring tools that we had, but basically the notes are, if there was a severe drought 1 to 2 years before, then it was quite likely that you would have a very strong nesting year the next year or in the given year, if drought happened 1 or 2 years before. So it's not the year of the drought that produced high nesting, it was preceded by drought. A year of good nesting is usually preceded by drought. So then this opened a couple hypotheses. Now, all I've written here is the observation, but the hypothesis that they came up with was that perhaps drought conditions are enhancing prey animal production, in the years after the drought. And they had two ways of thinking about that, but let's just clarify what we're talking about here. In other words, looking back a year from the nesting season to sort of the end of the previous nesting season, if conditions were really low in that previous nesting season, in other words, this black line reaches down to 0 or very close to 0 or even below 0, drying out the Everglades, it more than likely produced a very large nesting event that next year. So their mechanistic reasoning, or their reasoning for how this could possibly produce more wading bird food, went along one of two lines. First, drought or low water, and here's a little food web, a little food chain here, predatory fish, small prey animals, and algae. Drought could either reduce predatory fish by pathway 1, therefore releasing small prey animals, and indirectly, drought then produces more prey animals, or drought might enhance nutrient availability for algae and other resources, enhancing small prey animal growth rates, which would also increase small prey animal production, and together something increasing some animal here would then encourage more wading bird breeding. Okay, now I'm going to simplify this for the sake of the talk and move a little faster along, and we're going to put crayfish right in the middle here, and we're going to kind of ignore pathway 2, though we've studied that one a little bit as well. Well, what do we know about the Everglades? Well, we know hydrologic droughts act like classic ecological disturbances. Now, what does that mean? An ecological disturbance, by definition, isn't just something that changes something. An ecological disturbance is used by community ecologists or population ecologists, is an event that removes biomass and makes resources available. Well, in the Everglades, a big flat wetland like this, when it dries, I mean topographically flat wetland, historically no canals, remember, when it dries, the fish have nowhere to go, and the fish basically depend on water. So these fish at the bottom, which are our aquatic predators that feed on crayfish, for the most part die or have to migrate many, many kilometers to flee that drought. There are a few little alligators holes around, but they're extremely rare on the scale of the whole Everglades. They cannot support all the fish biomass that's out there. So what you get is a temporary loss of these fish. And that can last for months or a year or longer. At least reductions in those fish, I'll say. What's different about crayfish is that they can burrow. And you've probably noticed this. You've spent time outdoors, and perhaps you spent time outdoors around wetlands, and you notice these little burrows and chimneys. And the crayfish, their migration strategy or way of dealing with this is just to go about 10 to 20 or 30 or 40 centimeters below ground if the sediment is conducive to burrowing, and then they come back up. And when they come back up, when the water comes back up and they come back out, they find an environment devoid of predators. So that's the mechanistic reasoning for why crayfish could be conceptually involved in this drought-induced increase in production. So I want to talk about what we know, what we've known about that. I'm just going to kind of quickly move through about a handful of studies here that suggest that this is not a bad way of thinking about the Everglades at one level. So what we knew already before we started this work is that dry disturbances, or the drying out of a sediment surface in the Everglades, if it happens extensively, will reduce predatory fish populations. So I'm going to now talk a little bit about how dangerous predatory fish populations can be for crayfish production. And then we're going to ask whether dry disturbances really are correlated with crayfish density in the Everglades. So the first, we're going to move down to an experimental system. The University of Florida IFAS facility that I took advantage of for a couple of years in Davie, where we made these large mesocosms look like an Everglades Eleocharis marsh. And now we wanted to study the effects of these small, relatively small, 5 centimeter long fish predators. The dollar sunfish is on the top, the warmouth sunfish is on the bottom. And we were studying the effects of these two species, or combinations or densities of these animals, on production of the slough crayfish. Now, a crayfish, unlike a crab or a saltwater decapod crustacean, has direct development. So it starts out looking just like mom, only smaller. So this little tiny one on the bottom here is probably no longer than the width of your pinky nail, and actually starts out a little smaller than that. But then they go through a sequence of molts Somebody needs to turn something off there. Thank you. So they go through a series of molts, and then they grow up to sizes that the ibis actually want to feed on, and other large predators like maybe young alligators as well would feed on. But when they start, notice how much smaller they are than when they become ibis food. At this stage, they're dealing with all kinds of different predators, and these fish are just deadly, as well as even smaller predators could eat them as well. So what we did in this experiment is we basically stock different numbers of predatory fish in here to simulate the conditions that happen either long after the last drought, an inundated system for a long time, or a recent drought, so low densities or zero densities of these fish. And then my student came in and he stocked little baby crayfish in. He hatched them in the lab, and he stocked them in at night and seeded every one of these wetlands, a bunch of replicated wetlands, with little baby crayfish. And then we came back 12 weeks later to see what we found. And indeed, if there are no sunfish in the mesocosm, then you get a lot more crayfish biomass per meter squared coming out of these mesocosms. And these are sort of averages, and then some error variation around that. So if you have 15 fish, which is still less than 1 per meter squared of these fish in there, production is down, I don't know what that is, it's like 6-fold or something like that. Okay, so at a small scale, these fish are really dangerous for crayfish reproduction over a 12-week period in the summer. Even in a highly structured environment, there was vegetation, there were other food sources for those fish, there were shrimp and other fish and all kinds of things, dragonflies in there for them to eat. So it's not like we just fed these things to the fish. Now, we scaled that up then in 2009 to 11 with a study that we did at the Loxahatchee Impoundment Landscape Assessment. So if you know Loxahatchee National Wildlife Refuge, and I'm sure many of you do, these are the impoundments that were sort of rebuilt by the South Florida Water Management District on loan from the federal government for Everglades research. And you've perhaps walked around these impoundments looking for birds, many of you. And so we basically did a scaled up experiment of what I did in those small mesocosms. But in this case, what we did is we tracked the abundances of small fish and crayfish and predatory fish over a 2-year period in all 4 of these mesocosms, or macrocosms, we'll call them. They're bigger, they're macro. And then we dried 2 of them out in between the 2 years and reduced the fish predatory fish densities in those 2. And so we have 2 of them that stayed wet, and the predatory fish stayed intact. And in 2 of them, we simulated a drought condition in the Everglades to see what happened to the crayfish production over the next year. So just a little bit of data from this. The 2 dried lines are the 2 macrocosms that are red here. And the 2 lines are showing the predatory fish catch on the y-axis through time over the 2 years. And in between, the dashed line just shows the period when we briefly dried the system out and removed fish and got the fish out. The crayfish, of course, were forced below ground. Small fish could live in some very small puddles that were remaining, some small alligator holes. The controls are in blue, and you can see the year before we dried them, they all have about the same densities of predatory fish. And in the year afterwards, we actually, what I've circled here with the ellipse, shows the 40 to 90% reduction in the predatory fish. Now, this was actually a very modest reduction in predatory fish compared to what happens in the Everglades. Fish are reduced by drought. So we think this is a fairly conservative, so to speak, test of predatory fish reduction. And again, our predators are these fish that are bigger than 5 centimeters. That's their standard length. And standard length on a fish is just the length between the nose and the meat, which is about right there. Those are fish that we know will eat juvenile crayfish. Okay. Now, how did we measure crayfish and fish abundances? For the big fish, we used nets that measured them overnight. And we're measuring their activity densities, or their abundance, relatively speaking. But for the small fishes and the crayfishes, we use throw trap. This is what we do a lot of in my lab at FIU. We're all over the system, measuring many times per year, measuring abundances of fish and crayfish across the Everglades in all kinds of different habitats. But essentially, this throw trap is a box it's open on the top and the bottom. It's thrown into the marsh from about a couple meters away. It falls down in. It's got a copper frame and mesh on all sides. It's a 1 millimeter mesh. And then we use this seine that's leaning against the trap here to scoop out all the fish and the crayfish and the shrimp. Then we do that multiple times and we get a measurement of density. per unit area for some sampled habitat. So we did this at Lila in all those wetlands before and after we dried them and removed the fish. Lo and behold, the slough crayfish, again, grows by about 3 times. So here's the dried wetlands, the same as the control wetlands in the year before drying. And the year after drying, they increase about 3-fold. On average, the densities are about 3-fold higher. Incidentally, we can measure predation rates by other methods and show that the predation rates on baby crayfish were higher in the control wetlands than the dried wetlands in this year after we dried the wetlands. So we really did remove fish, and it really did have an effect on a population that we were not seeding in, who was already there, and they just experienced much greater population growth. So we have about 3 times the crayfish production in one wetland, pair of wetlands, than the other pair in this experiment. And that was a pretty strong demonstration. And so we've demonstrated this twice experimentally, but what you really want to know is, can we detect the effect of drought in the natural system? And this is sort of an important final check on this idea that drought may, at least temporarily, enhance crayfish production by removing large predatory fishes. And we did this by using a 7-year time series of data that we had collected, again with a throw trap, from 8 sites here in Central Water Conservation Area 3A. And as part of that work, we measured density twice per year for 7 years at these 8 sites, and they span a bit of a range of hydro patterns. So some of them dry a little bit more than others, but all of these sites dried at some point in the 7 years. So we have the experience of seeing all of them after a dry condition and all of them after some wet conditions too. What we did is basically build a statistical model that just asked whether, among other things, but in particular, this was our hypothesis, that this period of time here, these are 2 different hydrographs or hydro patterns for 2 different of the wetlands, and these are centimeters water depth through time. Notice that some of them go below 0, and some of them, the one that's indicated by the red, goes below 0 more often, but they all go below 0 for some period of time. Now, these are the dry disturbances, or the length of days that the site went dry in the last dry season. So we used that as a variable to try and explain crayfish density, and lo and behold, crayfish density rises as a function of hydrologic disturbance or days dry in the previous year. Not only that, the relationship is clearest by the next dry season, or the next January at least, so that after a year of growing and trying to survive out there, these baby crayfish are actually achieving about 3-fold higher density if the site's dried for 60 to 80 days than if it was continuously wet. Continuous wet supports lots of fish. Okay, so we see both in the field pattern and the experiments the same basic result, that dry disturbances are correlated with crayfish density. We know they reduce predatory fish populations. We know predatory fish populations reduce crayfish by eating them. And so we have pretty good evidence for this part of our working model. And that seems to work pretty well for this species, the slough crayfish. Now, that helps us answer this uncertainty as of 2009 that Frederick brought to the attention of the community. And we know to some degree that the ibis where they nest today feed on crayfish fairly heavily in the central northern Everglades. And that we know that there is an important interplay of flood, or at least drought sequences, and the transient release of crayfish, or temporary release of crayfish populations in the central Everglades for that species of crayfish. Now, there's important uncertainty though as of 2017, and I've changed this now to hydrologic restoration now because there's some important things that are related to the restoration here that we need to think about. And the first is, how do we get white ibis nesting near the coast in the southern Everglades? Because today the nesting is further up into the middle of the Everglades, central Everglades and northern. And second, I've just told you that we need to dry out the Everglades to make more crayfish, to make more ibis, and obviously that's not going to fly because the Everglades was clearly wetter. So how did a wetter Everglades make more ibis if you need dry conditions to make more crayfish that they eat? So hopefully you see the enigma and the confusion that we still were left with. So we'll go to the next chapter in this book report. The question then is, what conditions enhance white ibis nesting in coastal colonies? Now, coastal colonies differ in a couple important ways from the colonies that we had been studying. One is that if we start looking at this map again of the coastal colonies, I've told you about one particular species of crayfish, the slough crayfish, which lives in central and northern Everglades, the dominant crayfish there. And right down the middle of Shark River Slough, it's also a dominant crayfish or dominant part of the biota there, along with a whole bunch of small fishes. But there's a second species of crayfish in the Everglades, which is the Everglades crayfish. The Everglades crayfish is a stronger burrower, faster grower, lives in short hydroperiod environments in the southern Everglades. It can live in wetlands that have 4 months of water, 3 months of water even, although they aren't that abundant in those wetlands, but they live in a sense up gradient elevationally, though it's a very slight gradient. They live up gradient from the slough crayfish, and we've got a series of papers on this as well, but I'm gonna skip ahead to the rest of the story now. So is this species though perhaps involved the nesting or the prey use dynamics of white ibis is then the question. Prey use in the ecotone colonies had not been studied since Kirchland's work in the 1970s. And so we really didn't know what supported them there. But when he did his work in the 1970s, the colonies had already been quite diminished by the drainage of the Everglades. But fish and crabs dominated the boluses or the food regurgitates that they found from chicks in those parts of the system. And so nothing had been done really since the 1970s on this question. So we did some work on this for 2 years, in 2017 and 2018. And when we started the study, I thought, well, this sounds like a fine and interesting study. I can't imagine we're going to find anything very different than what we found, but we'll go ahead and we're going to try this study and let's see what we find. Maybe we'll find they're eating crabs or something like that is what I thought. Providentially, Hurricane Irma changed the story here a little bit because it dumped a lot of water on Florida. And in September 2017, we saw Hurricane Irma come over and dump a lot of water, particularly on southwestern Florida, and flood this wetland, part of the wetland system in a way that we have hardly seen for quite some time, and certainly not been watching to see. So we got 2 very different years hydrologically in this study. Now, what this meant, though, and this is the part of the big story here, is that we had a return of large nesting colonies of white ibis to southern Everglades, along with a number of other species that nested alongside of them, including storks. This is the record from the wading bird reports of those 7 colonies in the southwestern portion of the Everglades since 1999.
Wow.
In 2018, we saw the largest recorded nesting event of white ibis in 87 years. And these are the 2 years that we happen to get the diet data from. And so we had a pretty nice contrast in terms of the response of the birds to the system. And hydrologically, we'll come back to that in a minute, exactly what the hydrology did look like, or the hydropattern looked like in these 2 years. Okay, so these are the colonies that we were studying. Here's the map of Everglades National Park, and the 3 colonies we managed to get down to. These are very remote areas that the district was kind enough to take us to in a helicopter, because there's really no other way to approach these places in southern Florida. But Cabbage Bay, Broad River Colony, and Otter Creek were the 3 that were principally active with ibis in these 2 years. In the first year, we studied Cabbage Bay and Otter Creek, and these are the number of nests that were estimated to be at each one of these colonies. And these are the number of food boluses or regurgitates that we were able to collect while in the colonies over a period of about 4 or 5 weeks. So it was very difficult to get them climbing through the mangroves to get these regurgitations from some of the chicks. And but that allowed us to reconstruct the diets of those birds at these 2 colonies. The next year, though, Cabbage Bay still nested, though it was a little bit earlier. There were a lot of nests, but Broad River also was active that year, and it also was an extremely large colony, and that's the one we studied in the second year of the study, which had 120. We collected 120 regurgitates over about 4 different visits to the colony over about a little over a month. So I'm going to show a little data on what we found in the diets. And here we are after the helicopter drops us off, trying to make our way into these colonies, and really going through the creeks was almost the only way to get to them. The helicopter would drop us. We'd have to walk into these colonies. And yes, there were alligators sometimes in these places. alligators. I think these alligators have never seen humans. But these are some of the birds nesting down in here. And I think these are mostly either egrets or storks on this corner, this white mass here. And then, but all through here, there are birds, the white dots all through here. And then there were a lot of ibis in the top of this picture that you're sort of off the screen. And many of them were nesting on the ground. So here are a couple ibis chicks, and this is basically what we would get out of a chick, gently massaging it. Often they would throw up voluntarily because they were a little nervous when we'd start walking by, and then we would feed them a little fresh dead crayfish that we'd frozen and thawed as a real replacement meal, because we wanted to know what they were eating, but we really didn't want to do them any harm. So we would take this back to the lab, and then we would dissect it. Now, what's interesting about this, if you look at that, which is kind of gross to look at, but it is a mash, and that's just what white ibis do to their prey when after they feed on it, it gets mashed before they regurgitate it to their young. And so it literally becomes a jigsaw puzzle of things to look through. So we took the straightforward approach of just pulling these pieces apart and trying to learn what these pieces are and what you can find in these boluses. And this is similar to what I showed earlier about the Loxahatchee diets, is we'd find crayfish rostrums, which is part of the carapace right between the eyes of the crayfish, crab claws in these boluses, insect heads, and then fish otoliths, which are just headstones out of the heads of fish. And these are little records of the fish that they almost like they have tree ring lines on them that tell you how fast the fish is growing. And the fish, it's like a part of their skull. And these you can take out of the skulls that you find in the boluses, and they have particular shapes to particular species of fish. So we could identify the species, and then by using a lot of size to size relationships and part to whole relationships, we could basically reconstruct prey biomass composition of each one of the regurgitations that we collected, and then estimate how much biomass of each prey type, large groups of prey, were in these diets. We could also do some work to identify the type of crayfish that was in the guts. Okay, so here's the way the data looked from the 2 years, and a pretty strong contrast between the 2 years. In 2017, when the colonies were small, they fed on mostly crabs at both of those small colonies. And then in 2018, a whole bunch of crayfish, and actually fairly good number of fish. And I'm going to talk about that a little bit more in just a moment here. But notice a lot of other prey in here that are not particularly important. Shrimp are not particularly important. Aquatic insects are sort of a minor part of diets of these ibises as well. So the total biomass per bolus, total prey added up all together, was also about double in 2018. So not only we have 30-fold more birds, they're feeding their chicks twice as big of a bolus, and it's a shift from feeding on salty crabs to crayfish and fish. Okay, as you look through time over the nesting season, this is kind of interesting and important to think about. What kicked off all this nesting? In other words, what were the birds looking at that they thought to themselves, hey, in 30 days, this is going to be still great to feed my young. So let's start right here, honey, and with our nest, and let's raise some chicks. Well, at the beginning of the nesting season, crayfish really dominated the boluses. In fact, these are very large boluses, very heavy, dominated by crayfish. And based on the way the system dries down, we can only presume that the only thing that makes any sense is that the pre-nesting foraging by these birds had to be pretty much crayfish as well, leading up to laying your eggs or while you got eggs. But this is the first data we could get on chicks fed. At any rate, the diet does shift a little bit as the season goes on, and they start to feed on more fish. And then if we look at the composition of the type of crayfish that's here, the two types of crayfish, we also can identify based on reproductive organs that at the very beginning, they were mostly this Everglades crayfish, P. alleni, and then they switched to P. phalanx as the season goes on. And this sort of makes sense. In other words, let's come back to this picture now. Here's the colony and the star. And they start by feeding on Everglades crayfish, which is the species that lives in these short hydroperiod wetlands that had been flooded quite deeply in 2018. And as the system starts to dry down and these habitats become less and less available, they move down into Shark River Slough and start feeding on Brockenbarus phalanx as well as fish. So I think we understand based on these diet analyses basically what the birds were doing, with a few questions remaining, and a few little, there's always more questions than answers, it seems like, but I think that we have at least a basic sequence that makes sense given this nesting. And in subsequent years, Mark Cook has been up over this part of the system and seen a lot of bird foraging in exactly this habitat here. This is Loop Road, is right here. So the south of Loop Road, these Lossman Slough, south of Loop Road, and a whole lot of ibis foraging right in that area. Okay, so were there more crayfish available in 2018? Over the population's bigger. We have a little bit of data on this from one of our colleagues at Everglades National Park who shared some data with us. These are not from throw traps, but from a different kind of trapping technique from some stations that extended into Shark River Slough and then up into the Marl Prairies up here where the Everglades crayfish is found. And indeed, there were more crayfish per sample site where he was trapping crayfish in 2018 than there were in 2017. Or these are actually data preceding the nesting season, so not exactly when they were feeding, but preceding it in each case. So was more foraging habitat available for these birds to feed in 2018? Well, I've already told you the hydro patterns differed a lot. And so in short, the answer is yes, about 4.5-fold more habitat became available for foraging ibis in the second year than the first year. And that was primarily due to an already wet year then made super wet by Hurricane Irma. So if we look and we grid up this habitat and use a hydrological model that the USGS, US Geological Survey, has available to us, and then we ask during this nesting season or at the initiation of this nesting season, what were the conditions for foraging that the ibis were looking at? In other words, how wet did this system look? Did it look good? Did it not look good? How wet was it? How dry was it? And where were the wet and dry spots relative to their nesting colonies each year? We can look at those using their data viewers, and you can see conditions that range from too wet for foraging to too dry for foraging for typical wading birds. And you can see that in the first year, in 2017, well, there was a lot of really good habitat right around these colonies. Look at all this green and light blue, which is about to become green. But notice also a lot of brown. In other words, this is dry land that they cannot forage on any longer. The fish have either moved on, the crayfish have moved on or gone below ground. So all this brown is unusable. It can't become available with further drying. It will only stay brown. So when nests were initiated, they were looking at a very dry landscape in 2017. And in contrast, in 2018, when nests were initiated, they actually were initiated earlier under even wetter conditions, but the system was very wet. The best habitat was further away from the colonies. Here's Broad River, and look how much further the green is from it. But look at all the potential with further drying to make good habitat when these habitats start to dry out. Notice also that there was wet conditions up here, perhaps a little bit on the too wet side, right up here. This is actually when they laid eggs in these habitats where we know the Everglades crayfish is. All right, so just coming back to this then, the production of prey populations, for at least for the crayfish and for this particular wading bird, seem to require, the peak here seems to matter in some habitats, and the dry condition back here seems to matter in some habitats. Now, how did these 2 years compare to each other, 2017 and 2018 in the Southern Everglades? So again, if we kind of go to one place in the landscape and we were to pick up a water gauge right here and just say, how do the conditions compare between the 2 years? What would we see? Well, 2017 and 2018, 17 in blue and 18 in orange. Notice where 0 is on the y-axis. In 2018, the system dried appropriately, which gets rid of the predatory fish. In 2017, it actually didn't and was probably harboring more fish, even up in those habitats that we consider seasonally drying wetlands. So that was a first sort of bad point for 2017. But then notice how wet 2018 gets. And here's Hurricane Irma, the spike. And then, but because of Irma and all the water that was in the landscape moving across the landscape, it kept it quite high. Basically, you gain an extra month of hydro period in these wetlands. And that means even shallower wetlands would have been wet that year and potentially more productive as well. So we have a longer wet season, so to speak, but a better dry season in those habitats where the crayfish are being produced. All right, so what have we learned? The coastal ecotone can still support large colonies of white ibis. That's point number one, though we hadn't seen it in 87 years until 2018. Two, the white ibis depend on freshwater prey, particularly crayfish, particularly to kick off nesting. They will switch over and use fish in some cases, and the other species of crayfish, but the Everglades crayfish seems to be important. And that in particular on the west side of Shark River Slough in 2018. 2018 had both better antecedent drying and greater flooding than 2017. So I want to put this together in one last graph, and then I'm going to ask for questions. And I know it's getting dark on my screen. That's because the sun's going down. I don't have any lights on in here, but I'll turn a light on in just a minute too. But let's think about this and think about the Everglades with me. Bear with me for one more maybe challenging slide. But think about the performance of a predator and prey on a single axis here, and our predator being our large fish, which eat up our crayfish so that the birds can't get them. And the crayfish being the prey. There's a general theory in ecology that disturbance or the frequency of disturbance should affect the strength of this predator-prey interaction so that the predators, if they're sensitive to that disturbance, become less and less abundant and less effective the stronger the disturbance gets. So if on the x-axis, we think about these places as places in the marsh, in the Everglades that experience either a high frequency of disturbance, meaning they're very short hydro periods, or a low frequency of disturbance, meaning they're basically lakes that never dry out, we could think about the predation pressure from the large fish is basically declining, monotonically, so to speak, or just directly declining, fewer and fewer big fish. Now, on the one hand, if you have an occasional dry event, you could increase crayfish production in these habitats on the left just by an occasional dry event. That's what we showed in the first part of the study, that you have transient reductions in fish by an occasional drought. Maybe 60 days dry might make more crayfish by reducing fish densities. On the other side of this, though, we have over-dried habitats that are also, just because they have very little water, maybe 4, 5, 6 months, will not produce a lot of crayfish because the crayfish are limited by water themselves. And so the flooding that we see in the Southern Everglades in 2018, combined with a seasonal drought that gets rid of the fish, produces also a movement in this direction from the right to the left by flooding. So you're making a very dry habitat a little wetter, or you make a very wet habitat a little drier occasionally. And so either modest drying of sloughs can make more crayfish, or flooding of seasonal prairies. This argues for a big Everglades with a diversity of habitats that many of which will have at least on the periphery in the Southern Everglades, have seasonal drying, but have more water in the wet season. Okay. So in summary, crayfish populations might be enhanced by drought or flood, depending on the initial condition. But white ibis throughout the system seems to rely on crayfish during breeding. And so I think we've moved quite a ways in our understanding of hydrologic variation and white ibis breeding activity. And hopefully, through adaptive management and future restoration plans, we can make some of these interactions a reality to increase the eruptive breeding of white ibis breeding along the coast and bring more breeding back to the coast of Everglades National Park. Okay, and now I will take questions.
Hey, thank you. That was great.
Hold on,
I
let me
had,
light.
yeah, go for it. I had so many questions that my brain shut down. But I will read you a couple of the questions that came in while you were talking. And the first one is just, Marie asks if the Everglades crayfish are they a strict freshwater species?
Yes, they are strictly freshwater as far as we know. However, I will say, without trying to be long-winded, that the freshwater conditions in the Everglades historically reached right up to what we think of as the coast today. So there was a very narrow estuary. And there are fish that you can still find, like a red drum, you can find with crayfish in their guts. It's a saltwater fish or an estuarine fish, but you can find crayfish in their guts. So they will access these crayfish. But as far as we know, the salinity limits of these crayfish, they don't have a lot of they don't have a lot of sensitivity. They're not tolerant to much salinity. That's a better way to say it.
Okay. So in general, they're strict, they're a freshwater.
Yes, yeah, you're not gonna find them in the estuary.
Yeah. And Karen wants to know, so should we be promoting crayfish / crabs in HOA communities by the ocean? Because they see a lot of ibis there.
So it's kind of interesting. You know, so what I just said, my sons want to know this too, like the lake behind my house, you know, can we get more crayfish in here, Dad? And I said, well, sons, I think that there's just too many fish. Without a big flat structured wetland habitat, you're not going to get a lot of crayfish. What you're seeing, of course, in HOA communities is there's both, you know, feeding of ibis, but there's also going to be a lot of use of the lawns. This is also something we did pick up studying the diets as extensively as we did, is that they're feeding mostly on the Suriname cockroach in your lawn. And the Suriname cockroaches move around in your lawn after rains. And that's why you see the ibis pour in after rains. The water for the cockroach moves them in the opposite direction. They're trying to get up out of being flooded. So they start moving around with rain, and the ibis come in and can find them more easily. So they're taking advantage of the large number of lawns and cockroach production. Now, the question though is, does that support nesting?
Yeah.
And we don't see that. And if Audubon wants to help us find the nesting colonies that are in the urban environment, that would be great, but we don't see them. And if someone wants to tip us off, not just that there are rookeries, but nesting colonies that are being supported by garbage or roaches and lawns. Please tell Mark Cook where they are, or tell me, or anybody who's studying this stuff, where these colonies are. We know they use them during nesting, but they seem to be a secondary resource.
Yeah. So there you go, Karen. Yeah. Go find those.
Fish. Yeah.
Yeah.
If
So
you can
Karen,
get
go
them-
find those guys.
Yeah.
Okay, let's see. No more. So who else does anybody else have a question? We don't have any more questions coming in right now. Except for Jeanette says oh, this is really interesting. How do you define garbage?
That's a good question. And if you want to get really technical, I think my co-authors wished I hadn't used that word, refuse. So this is human waste primarily. Well, I can't say it's human waste, but these are birds that are either the contents of the garbage category is maybe the easier way to say what the definition was in our study was what we know, dog food, chicken, like chicken breast,
Yeah.
Tofu. So those are things that were strong enough to last in the gut that we could identify it. And so we did pick up a little bit of that, but particularly in a year where the wetland got so dry, there was no wetland left available to feed in. So
there you go. You've got a lot of compliments coming in. Oh, will they eat commercial bird food?
that's a good question. I don't believe so, but I don't know. I remember we're talking about a tactile forager, and its principal prey is going to be about an inch long, and they feel their prey. They don't see it, and something I just briefly mentioned, but so I think they'll pick up lots of things, but if it doesn't feel like food, they generally won't eat it. Although we've found maybe seeds in their diet once in a while, but big seeds, really big seeds.
This is a good one from Ron. If an urban garbage area is identified, garbage, can the birds be tagged to identify their nesting areas?
So there's a group from Georgia that's kind of working on this question of the urban ibis. And so that you can take this question from one of two directions, and they started with it, trying to understand, how healthy or unhealthy are the ibis in South Florida, but they are doing some tagging. And the last study I saw, the ibis, once the breeding season's over and the water levels start to go up in the wetlands, they can no longer use the wetlands. So they will spread back all over Florida or the southeastern US. Some will go to the coast and they'll feed on crabs. And I didn't mention this. Let me back up a second. Crabs are acceptable food for ibis adults. Crabs, it appears, physiologically are bad food for ibis chicks because they can't get rid of the salt and they don't have a source of fresh water to dilute it. So they actually, the one physiological study that was done back in the 90s showed that they lost weight when fed crabs.
Huh.
But the adults, once the season's over and the young of year, will spread to the coast, and they'll spread anywhere where they think they can get a decent meal. Some of that may be garbage dumps. Some of that may be your lawn. Some of that's your neighbor's backyard who feeds bread to the ducks. And some of it's going to be up into Georgia. So this large population of breeding birds sort of nomadically goes all over the place after the season's over. They leave the Everglades, except maybe some of the margins of the Everglades they may still use. And then they'll come back to it. Many of them, tens of thousands, or maybe 10,000 in a bad year, and 80,000 in an excellent year in the winter. So I think people are tagging them. The last data I saw suggested that the birds still do annually go out into the Everglades for nesting. So, I think that's a really important point. I think the idea is getting out there that these are urban birds.
Mhm.
And we have to be careful about how we define urban bird. Is it a bird that could we remove the wetland and have a viable population of white ibis in the world?
Right.
And I don't think that we see that would be possible. Although I know they take advantage of some urban resources seasonally. So.
Right. Cool. And someone is asking again, what do they eat in lawns, which is back to the Suriname cockroaches.
Yeah, they eat several things in lawns. They will eat worms, earthworms as well, and mole crickets. But what we don't see lots of toads or lizards like you might find in a great egret or something like they eat lizards, I think. And but mostly it's those invertebrates. And the Suriname cockroach, at least in Palm Beach and Broward County, seems to be a big one.
Ooh. So this is a question kind of about what you were just saying. They see juvenile white ibis in Lake Worth, so were they born in the Everglades? And
yes, almost certainly born in the Everglades. So there are colonies of white ibis. I don't know that there have been any recent colonies in the Kissimmee River floodplain, but they will nest in the littoral zones of Lake Okeechobee too. And those colonies will eat a little bit more fish, still eat crayfish, and they will feed in the agricultural lands around Lake Okeechobee. But those tend to be pretty small colonies, maybe 1,000 at the most pairs, but 300, 400 birds or pairs, I should say, nests. So where exactly they were born, it could be anywhere from Lake Okeechobee down to Everglades National Park, Broadus Pond, or something like that.
cool. Great. Let's see. I think that was the last question. And oh wait, here's somebody. Scott. What? Scott says, compared to roaches or crabs or other food sources, are crayfish more protein nutrient dense?
I don't know if we know that. I know that so how to measure the quality of a food prey item is a really important question. And ecologists and I think even avian ecologists that I've been reading about now are thinking about food quality in a variety of ways. And I actually asked this question of some of my really great avian ecologist colleagues, and I never really got a clear answer on that one, but we've done a little work energetically on this. And a fish energetically is a little better than a crayfish, gram per gram. But the question is it energetics that the birds are struggling to get? Or as people are talking about today a little more, they need fatty acids. Or is it protein? Or for the birds even, or for the birds eating crayfish, is there something else? Is it need carotenoids? I don't know, because crayfish are full of carotenoids. Like you think about flamingos feeding on pink shrimp, and that's how they get their pink color. Well, the crayfish are certainly being fed on when they at least part of their diet when they color up the males and the females. So how important is that for their, I don't know, but we've not compared crabs directly to crayfish. I would guess crayfish are probably more energetically favorable than crabs because of their carapace is so much more thin. So gram for gram, you're getting more protein. And we've done the cockroaches too, and I want to say they're close to a crayfish, but terrestrial animals tend to not have as many of the essential fatty acids that heterotrophs need. That's I'm speaking generally, not specifically about the cockroach. So my guess would be for fatty acids, the crayfish may be better too.
I think it's really interesting to me how much with a bird that you see so much, like how much we don't know. It's that like really is, I don't know why that interests me a lot, that like that those unknowns always fascinate me. Anyway, somebody says, Ron says, people feeding urban ibises garbage are violating FCW law. Is there communication between you and FWC?
No, not right now. There isn't. I can't say that. I can't say that my I have a neighbor I'm looking at right now across the lake who he's all out. But I think that that's challenging because we don't. I think people who are working on this question in parks, may want to make those connections with FWC, and likely they've got the permits to do it. They've got the permits anyway from FWC to work on those birds. They should be pointing it out. I've mostly worked on ibis at the colony. And I think some of the urban feeding is accidental. I mean, the ibis are aggressive. They'll take it from the ducks. Somebody's not targeting ibis necessarily. It's just a free meal for the ibis, and it isn't very valuable, but it's a free meal. I think there was a question about the green K.
Yeah, that was the next one, Frederick.
And I've not been up there. I know green K, I've been there, but it's been years. The question would be, are these rookeries or nesting colonies? And then secondarily, you'd have to obviously go and see, because you're not too far from Loxahatchee right there. There are storks that are using urban substrates to nest in and on islands surrounded by a lake, which is a safe spot to get away from raccoons. But then some of their feeding might be in urban type wetlands, and then some of their feeding for a stork, because they can fly a lot further, they're still going to the Everglades, but they're using an urban substrate to nest in. So there's a sort of a little series of questions that have to be asked there. But if it's a nesting colony, that's kind of an interesting observation. And again, like if we could let Mark Cook know, I'm sure he'd like to know, or Mike Baranski, who helps him with that wading bird report. I'd be fine to know too. But they keep on top of those things a little bit more than I do. I'm busy studying fish and crayfish.
Anthony wants to know if there's anything to mention in regard to the Burmese python. Like, did that affect anything on your study?
No, well, actually, I'm gonna take you back. Can I share the screen one more time?
Yeah.
All right. So this is for. Hold on a second here. Let me see if I can get there. If people can hang on just for 1 second. Okay. Okay, you can ignore this first screen here. Okay, so this is a picture of the ground nesting ibis from that big colony that we studied. And so you can see this is how we count. This is how Mark and colleagues, Lori Oberhofer at National Park, how we count ibis nesting. You have to get them basically when they're synchronously on nests, or you could never count the size of these colonies because they move around too much. And they're off foraging, and you would never know exactly how many chicks there are because you can't count the chicks, they're too dark. So I was looking through this one day and just looking at it and counting it, and then I came across this right here. And let me zoom in. And there's a python right
Okay.
there,
Yeah,
right there.
wow.
Took me a long time to find it. I didn't know it was going to be there, but we knew there was a python out there. And I think the last day of the study, when most of the chicks were up and off the nest, Mark and my student Tassa did come across a very large python strangling some chicks and eating them. I don't know that they're going to be able to have really strong measurable impacts on these colonies at this point. It's 5 weeks of a concentrated resource, and how much damage could even 20 snakes do to 10,000 pairs of ibis and all their chicks? I don't know.
Hmm.
It's hard to say, because those snakes have to be able to survive there the rest of the year when there's no ibis and other historic resource, right? So anyway, good question though.
Yeah, that was really interesting. And I think that was the last of our questions, which was perfect timing.
great.
And everybody, there's a ton of compliments coming in. So thank you so much. It was really great. I found it really interesting. And I'm going to throw it back over to Natasha to close up.
All right,
Thank you,
Autumn.
Autumn. Thank you, Dr. Dorn. That was an amazing presentation. I never would have spotted that python in a million years in that photo. So that's pretty amazing that you spotted it there. So with that, I'm going to go ahead and just remind everybody that our meeting next month is going to be on August 2nd. I will be your presenter. So if you guys want to hear about how STAs are created and kind of what my role is in that at the Water Management District, then make sure you register on the Zoom RSVP, and I'll try to make it fun and interactive if I have time. With that, thank you everyone for being here tonight. It was an amazing presentation, and we always look forward to seeing everybody's faces virtually. So until August 2nd, have a wonderful evening, everyone.
Thanks,
Thank you.
Natasha.
Thanks,
Thank you very
everyone.
much.